everybody. I'm going to have a presentation that I'm going to put up in just one second, but I wanted to introduce myself a little bit really quickly. My name is Katharine. You can call me Dr. Miller if you want to. You can also call me Katharine. We don't need to stand on formality. I received my undergraduate degree from Northwestern University in biology, and then I went on to get my PhD in Germany at the University of Hamburg, where I studied molecular neurobiology. So I studied how the molecules that cause neurodegenerative disease function. And after that, I did a postdoctoral position that took place partially in Germany and partially at UCSF in San Francisco, where we had a really cool chance to get to study how microgravity affects the development of cardiovascular disease. And we got to partner with NASA Ames in order to do that and send some mice up to the International Space Station in order to learn more about that. And I think that that's going to probably tie in a little bit to, I think, some of your projects today. I don't know. And now I'm doing a postdoctoral position at Stanford University, where I'm in the Department of Otolaryngology, which stands for ear and throat research. So I study the inner ear. I study hearing loss. And that's what I'm going to talk about a little bit today. I'm going to try and talk about it from sort of a broad concept of how hearing works. And then I'm going to talk a little bit about the type of research that I do, imaging techniques that I use, so that you can see what the inner ear actually looks like. So I know that all of you are very into science, and I hope that I have put this at the right level, but I also am very happy to make this interactive. So if any of you have questions as I'm going through, or if something I say doesn't make sense, please don't hesitate to jump off mute and ask me anything really about what I'm going to be talking about. So first of all, why do I do the research that I do? Who is affected by hearing loss? And the answer is that there are a lot of people, really anybody can be affected by hearing loss. 0.3% of newborns are born with hearing impediments. 16% of American adults, and 50% of adults over the age of 75 years. So it really affects a wide range of people. There are different types of hearing loss. There's something called congenital hearing loss, which is when you're born with a hearing loss. There's also something called progressive hearing loss, which would be something that develops over time. There is hearing loss that is genetic. And then there's also hearing loss that can be caused by an external event. And what I'm particularly interested in is the genetic forms of hearing loss, because these are things that we have the ability potentially in the near future to fix using gene therapy, which is something I'm sure most of you guys have heard or read about. So I study genetic forms of hearing loss. But before we really talk about what those might look like, let's talk about how hearing works. So I'm going to start a little bit with the physiology of hearing. So you guys, when you think of the ear, you think of what comes out of the side of your head, which is part of the whole deal. So that's called the pinna. That's the external part of the ear. You also have the ear canal, which comes to the tympanic membrane. It connects to these three little tiny bones, which you probably learned about in some of your biology courses. And those bones connect into what is known as the cochlea. And the cochlea is the main part of the inner ear. This is the part of the ear that I actually study. And I'm going to talk to you more about how that works. So sound waves are actually transferred to the cochlea. They come in through this external canal. They vibrate the tympanic membrane. And then when the tympanic membrane passes these vibrations through these bones into the cochlea, the waves then sort of circle through. I think you can see that if I play it one more time. They sort of circle through this, what looks almost like a snail shell. And then these impulses are passed on into the brain. So how the waves are registered by the cochlea before they are communicated through the nerves to the brain is actually a really fascinating area of study, I think. Of course I do, I study it. So if we zoom in on the cochlea, we have sort of two major things that are happening here. We have the cochlea, which is this snail shell spiral. And then over here, we have something called the semicircular canals. And the semicircular canals are part of what you maybe know as the vestibular system. I don't know if you've ever heard that term before, but basically the vestibular system is important for balance. So your inner ear does two things. One is hearing and the other is balance. And you can see that these semicircular canals, they go in three sort of different directions. And that's so that when you tilt your head in different directions, you're able to not fall over. Basically, it lets you know where you are relative in space to everything that's around you. And I do study that a bit, but I'm mostly focused on the cochlea. So you see this snail shell orientation. Now imagine if you took that and you unrolled it. You would have this very long sort of organ, and it's called an organ. I think of it actually as a piano, which is sort of a joke, but it's also true. We have cells that run along this long strip of organ that is spiraled up into the cochlea called hair cells. And if we slice through that long strip and look at it from that slice, so down through the middle, this is what it would look like. So you have lots of cells. It's actually quite thin. Lots of cells that are supporting. These are the light pink cells. They sort of are holding things together. And then we have something over the top called the tectorial membrane, which is called the TM here. And then we have these three cells in blue here and another blue cell over here. These are called the hair cells. And these are the really important cells involved in hearing. So you can imagine, I think, why they're called hair cells. If you look closely at these four cells, they have these little tiny hairs sticking out of the top. They're actually called stereocilia. They're not actually hairs, but they do look like hairs. And we'll see a lot more of these coming pretty soon because this is what I focus on. So we have two types of hair cells here. We have an inner hair cell, IHC, and 3 rows of outer hair cells. And these hair cells function differently. They have 2 different very important functions. The inner hair cells I think of as binary cells. If they are working, you can hear. If they are not working, you can't hear. However, if you maybe have some grandparents who are getting a little bit older and they can hear, but you have to shout for them to be able to hear you, that's because their outer hair cells are maybe not functioning. The outer hair cells are actually our amplification cells. And I'll show you a video of what that looks like later. But they actually have the ability to take a tone and amplify that tone. So they help the inner hair cells to not only recognize that sound is happening, but then to help you to hear quieter noises. So this is what it looks like when you slice through this organ that spirals through the cochlea. So if you take that organ and you unroll it, like I said, it's like a piano. These cells are running along, it's running along the cochlea from the bottom all the way up to the top. And these hair cells at each of these different points recognize different pitches of sound. So at the very top, you hear very low pitches. And then as it spirals down at the very bottom, you can hear high pitches. And there's a really great video that shows what this might look like if you were to listen to a piece of music. And I'm going to show you what that looks like. So here's our outer ear, and we're entering in through the external auditory canal, through the membrane. Here's the cochlea. And then if we unroll the cochlea, and we look down at the organ. And I hope the sound will work on this. Yep. All right, so you can sort of see how everything is sort of being triggered at different parts of the organ of Corti, this organ that is spiraling up into the cochlea. And what I really like about that video is as you're watching it, it's actually happening in real time in your ear. So it's kind of fun to get to see that visually. We don't often get to see things that are actually happening in our bodies as they are happening. So back to our hair cells. So as I said before, we have our inner hair cells and our outer hair cells. We have our amplification cells. Yep, there we go, labeled. And these bundles that are very important, these hair bundles, are deflected by the sound waves as they are pressing up against this membrane that sits over the organ. And when they are deflected, there's a tension that is put. Oh, sorry, go ahead. Was there a question?
No, I don't think so. I think someone's audio just turned on by
Oh, okay, sorry. Okay, so as these hair bundles are deflected, tension is put across little tiny links that connect these stereocilia together. And this tension opens up little tiny holes called channels in these bundles that allow chemical ions to flow into the cells. And when those ions flow into the cells and these cells are triggered, they depolarize and they send a message to the brain through the nerves. And this is how you recognize that you have heard a certain pitch. So again, if we watch this, we see the sound wave is deflecting these specific hair bundles, and we're causing this hair cell here to depolarize, and it's sending a message into the brain. So this is one of my favorite videos. So when I first started researching hearing loss, actually back when I was an undergraduate at Northwestern, I studied the outer hair cells. I now focus more on the inner hair cells. But the outer hair cells, as I said before, are the amplification cells. And they have a protein that is embedded into the outer membrane of the hair cells. So if you can see here, this is a real outer hair cell. It's been taken out of the rest of the organ. And in this membrane, there are just a ton of these little tiny proteins called Preston. Now, if any of you are musicians, you might recognize the term presto. So presto means very fast, right? So the name Preston, which was named by my former professor at Northwestern University, is because this protein is the fastest moving motor protein in the human body. And what that means is you have all of these motor proteins that when these cells are triggered, the cell jumps up and down. Quite literally, it bounces up and down to the rhythm of whatever pitch is triggering it. And so if I show you this video here, if you look closely, you're going to see this hair cell dance to the music that's being played for it. So hopefully this will work too.
We can't hear anything.
So you can see it bouncing up and down to the music. So that's, again, sorry, that was a little loud. Again, that's exactly how what's going on in your ear whenever you hear any sound, which I don't know if you guys can hear. You may hear my daughter going down for a nap in the background as well. So yes, you hear these, you see these outer hair cells bouncing up and down, amplifying the sounds so that the inner hair cells can pass the message into to our brains. So these hair bundles, like I said, are what I'm very interested in studying. So we've seen a lot of cartoon depictions of what this looks like. You see these little tiny links that connect the different stereocilia to these little tiny channels that are important to open, that let ions in, that pass the message to the brain. So how can we actually look at these hair bundles? And that's what I do. I try to find different ways of looking at these hair bundles. So I can skip that. So one of the ways that I look at these hair bundles is through something called fluorescent microscopy. So this is one of the most basic forms of microscopy that many, many people use in the labs. And the way that we can do this is by attaching something called a fluorophore. This is one way of doing it. So a fluorophore is something that fluoresces. Attaching a fluorophore to an antibody. So you have probably heard of antibodies as being in your body, and they recognize when something bad is attacking you, a bacteria or a virus, and they rally your immune cells to come and attack those other cells. The way antibodies actually work is they recognize specific proteins that are in these bacteria or viruses. So we can use that ability. We can create antibodies that are specific to different types of proteins that we are interested in looking at, and we attach a fluorophore to those antibodies, and then we can wash tissue with it, and we can take a look through our fluorescent microscope and see where is their fluorescence happening. So the way fluorescence microscopy works, and I think many of you probably already know this, but we have something called an excitation and an emission. So the fluorophore will be excited by a certain wavelength of light, and it will emit a second wavelength of light. So we have one wavelength of light, or a very small range of wavelength of light coming in. It hits our specimen. If we have the correct fluorophore there, that fluorophore will be excited and it will emit a different color of fluorescence. And then that goes through the detector. You can look through the microscope or you can take images. So these are some of my images on what the hair bundles in the inner ear actually look like when we're looking at fluorescent, using fluorescence microscopy. And you can see that this is actually a really powerful technique. We can look at different protein expressions. Over here, you can see proteins that are expressed exclusively at the very tops of the stereocilia. You can see proteins that maybe go up around the outside of the stereocilia, sort of encasing the internal portion of the stereocilia. This is an image over here. This same image, if you go down from the very top and you slice through over and over and over again, you can see that these proteins actually form little rings around the inside of these stereocilia. So it gives us a lot of variety in how we're able to see different types of protein expression in the hair bundles. And it gives us a high level of localization, which is important. So you have to understand where all of the different machinery parts are in order to understand what they're doing. The downside to fluorescence microscopy, because it's based on light, we are only able to get to a certain level of resolution. If we want to get to a higher level of resolution, we need to use something called electron microscopy. And this is actually a pretty complicated technique. There are two major types of electron microscopy. there's scanning electron microscopy and transmission electron microscopy. So you guys know electrons are going to be able to give you a much, much higher resolution than light, because electrons are going to be able to really get down to the nitty gritty. So scanning electron microscopy, basically similar to fluorescent microscopy, you are going to be bouncing your electrons off of your sample onto a detector. So we coat our sample with some type of metal, and then we bounce the electrons off of that, and it goes straight into a detector. Transmission electron microscopy, on the other hand, your electrons pass through the sample and onto a screen. And I'm going to show you why those are two good different techniques to be able to have. So starting with scanning electron microscopy, this is what the resolution that we can get looking at a hair bundle might look like. So you can see all of these different stereocilia. You can see all of the different shapes, the different heights. You can see what's happening around them. You can see these different tiny little connections that are happening between them. These different links that are very important for keeping the hair bundle intact. And actually, the shapes of these hair bundles will change under different forms of hearing loss. So you can see up at the top, this is what a normal hair bundle would look like. And then under 3 different types of genetic hearing loss, you can see that the hair bundle shapes, structures change pretty drastically. So this can tell us a lot about the types of hearing loss that we are researching. And we can zoom in really, really closely, and we can see these tiny little tip links that connect the tall rows of stereocilia to the shorter rows of stereocilia that open these gates. And this is the area that I'm particularly passionate about. I'm very interested in the tip links, how they function, what they're connected to, and how they're able to open these channels. So if we go back to the types of electron microscopy, so that was bouncing off. So we are able to see the external structures of what we're looking at. Transmission electron microscopy, we need a very, very thin sample. So we slice our samples super, super thin, and we pass electrons through. This allows us to see the internal structures of what's going on inside the stereocilia. So even higher resolution than scanning electron microscopy. And that's what this might look like. So this is a slice through a hair bundle. So we're able to see the insides of the hair bundles. You can see these little tiny lines running up and down inside of the stereocilia. These are some of our structural proteins. This dark area here, this is where the tip link actually inserts into the side of the stereocilia. And if you look really closely, you can actually see a little bit of the tip link coming off into the ether. Using this, we're actually able to see the internal structures that occur within the hair bundles. And again, we can see differences that happen between different types of genetic forms of hearing loss. And as we start to compile all of that information together with biochemistry, with cell culture research, and with behavioral research, we're able to understand the different molecules that are important for hearing and how they function differently or dysfunction or stop functioning in different types of hearing loss. So I hope that was at the right level for all of you. I know you guys are all very smart, so hopefully it wasn't too low, and hopefully I didn't overshoot either. I would love to hear if you guys have any questions about what I talked about or anything to do with science or hearing before we go into the breakout rooms, if we have time for that.
We can answer like a few questions and then we'll start presenting.
Great.
Thank you so much for your presentation. Okay. Sorry.
I have a question.
Yes.
So what drove you to study hearing loss and further to the hair cells?
So when I was an undergraduate, I knew that I wanted to study biology, and one of the laboratories that had an opening in it was a hearing research lab. And as you will learn if you follow this path, there are lots of different types of jobs for undergraduates, and some of the jobs for undergraduates are very simple jobs, and some of the jobs for undergraduates you really get into the science. And I knew I wanted to really get into the science. And so they offered me an opportunity where I would work during the school year part-time, and then over the summers full-time. And actually, after I graduated, I stayed on for a few years afterwards full-time. And we created this very beautiful story to understand how one of the proteins of the hair bundles works. And so that was my introduction to biological research. My PhD and my first postdoc were actually in different areas of research. And after a while, I said, you know what, I'm still really passionate about this area of research, and I decided to come back to it. So it's sort of a roundabout way, but it's a good question. Yep.
Anyone else have any questions?
I have a question.
Actually, oh yeah, you can go ahead.
Okay, thank you. My question is, what do you like to do during your free time?
Oh, well, normally I love to go for hikes. I have a 1-year-old daughter now, so it's a little bit harder at the moment, particularly with the pandemic. But I'm a big hiker, and the Bay Area is great for that. I also am a big reader. I love all types of books, but I'm particularly like science fiction and fantasy. So, yep, those are sort of the two big ones.
This is sort of unrelated to hearing, but Northwestern happens to be my dream school.
Yeah.
And I was just wondering what was it like there, and did you enjoy it and stuff like that?
Oh, I loved Northwestern. It was a fantastic school to go to. So Chicago is great. I mean, it has a very different climate from here, but it's a very bustling city, lots to do there. Northwestern itself is located right next to the lake, so it's this really beautiful area. The university has so many opportunities, both for courses where you can take courses, from many different departments, but also outside of that. And they really encourage you to get involved. And I was in a lot of groups. I was in the marching band. I was in the ballroom dance team and things like that. So it's a good opportunity to get to do lots of different things while you're focusing also on intense study. And I think it was a really great place to give me connections that I've used my entire life for my career and also personally. I'm still very, very good friends with the people who I met while I was there. So highly recommend Northwestern.